Welcome everyone. My name is Agnese Winfield from NanoString Marketing. It is really my pleasure to open today now the Spatial Answer Trilogy. There are 3 events. The first one today focused on oncology, second one tomorrow focused on immunology, and in today's time on Thursday we have the Spatial Answers focused on neuroscience research. So the event is sponsored by NanoString and Illumina and with the great scientific support of the EACR. Before I introduce our speaker and our chair, most of all, for today, I want to remind everybody that this is a live webinar. You can post, and we encourage you to post your questions on the button on the left of your screen, the question and answer box. And we'll answer them, and we'll strive to answer them all at the end of the presentations. As I said, we are live, but you can also watch this presentation recorded. As soon as we finish, it will be available on demand. Let me just introduce now our chair today, Professor Juan Seoane, Group Leader and Director of the Translational Research Program at the Vall d'Hebron Institute of Oncology and Professor of the Autonomous University of Barcelona. Juan will share this session today. He's the Secretary General of the EACR Association and will take us through the presentations and the question and answers and has a great announcement later on to make too. Over to you, Juan, now. Thank you.
All right. Thank you very much, Agnese. It's a fantastic opportunity to be in this session. I think it's going to be a fantastic session. But before starting this session today, I want to pre-announce that we are going to open a new spatial profiling grant today, and we will give you all the details after the speakers' presentations. So please stay tuned to the end, and you will learn about the whole transcriptome in situ EACR grant for your FFPE and fresh frozen samples. So today we will start with 2 short presentations, 5 minutes each, from Rudy from NanoString and Anna from Illumina, who will share some brief information about the technologies that are used by our presenters today. So, okay, let's go with the first one.
Thank you, Juan, for the nice introduction. So I've been given the difficult task to present to you, give you an introduction in spatial biology in just 5 minutes. So my name is Rudy van Eysden. I'm the manager of the product application scientist team at NanoString Technologies. Well, I'll get started explaining about spatial biology in 5 minutes. So if you would look at NanoString's website, look at the over 4,000 publications and do a search, you'll see that 1,700 publications are in some way related to the development or validation of signatures. So that's more than 40% of the total publication number. So people have been using the NanoString technologies in some way to discover and validate biomarker signatures and bring them into the clinic as well. Most of them is using bulk gene expression analysis. Sometimes even with great technology, it's difficult to find the right biomarkers. And this is illustrated very well with this publication, which is a collaboration with NanoString and Merck, where they have identified a gene expression score, a T-cell inflamed gene expression score, also known as the tumor information signature, which is associated with progression-free survival and also is somewhat predictive of response to treatment. However, in the plot on the right side, you see that there are quite a bunch of patients that have a high tumor information signature score, but still do not respond very well to treatment. And there are reasons for that. So why is that gene signature not performing very well? It's all due to heterogeneity of the tissue. So here on the left side, we see a hot tumor, and on the right side as well, because they both have high tumor information signature scores. However, on the left side, And on the right side, we see differences with respect to immune cell infiltration. And that could be the reason why one of these patients, typically the one on the left, is not responsive to treatment. So this is a very interesting aspect. So if you would do bulk gene expression analysis on this, you would not find those differences because it would all level out in the middle of this bulk grind and bind assay. It's also illustrated by this more schematically. If you have responders, non-responders, if you would do bulk, you get similar gene expression profiles. However, if we would allow you to focus on specific parts of the tissue, you might see differences, making it easier to find the right biomarkers. While doing that, looking at spatial information, it's always a trade-off. If you go spatial, you need to give in on plexing, so the number of targets that you look at. If you go higher plex, you very often have to give up on the spatial information. Secondly, there's always a trade-off in quantitation and precision. So we believe that with the NanoString Genomics Digital Spatial Profiler, we can make life a lot easier. And it has already been proven because at this point, there are already over 45 peer-reviewed publications, not in the least type of journals. We have some covers as well that you can see on the left side, Clinical Cancer Research, Nature Medicine, all the big journals as well. So let me explain you in a few slides how the technology works before I hand it over to the speakers that gives you an explanation how they use the technology in their research. So the technology is all based on either antibodies if you want to detect proteins, or in situ hybridization probes if you want to investigate gene expression with spatial information. In both situations, we attach a DSP barcode, which is photocleavable by using a photocleavable linker. So your UV light will make that DSP barcode cleave off. The barcode has a unique sequence, and there's one unique sequence for every individual target that we want to measure. So this is the schematic workflow. So first, there's always a staining of your FFPE or fresh frozen section. In that staining, you will have either in situ hybridization probes or antibodies labeled with the DSP barcode. On top of that, there will be fluorescent markers giving you the morphology of the tissue. That's step 2. Based on that morphology, you'll indicate which regions you want to analyze in more depth using the panel that you've chosen. And then we shine UV light specifically and only within those regions, cleaving off the DSP tags and collecting them in a 96-well collection plate. Then we repeat that for all the other regions of interest that you've selected. Then we'll take that collection plate, and then we can count the abundance levels of the DSP barcodes referring to the abundance levels of the targets by using either the NanoString Uncounter technology or the Illumina next generation sequencing for counting. In the end, for every region of interest on every sample, and then sometimes you can split up the regions of interest in different segments, different compartments within the tissue, you get digital counts presented in a table like that, which is then subsequently imported back into the Genomics DSP software, connecting the data back to the spatial location. So this way, you can detect different types of targets, RNA and protein. You can use it on fresh frozen and FFPE tissue. So you're really looking at the spatial biology of different tissue types if you want. And it's optimized, it's proven by all the many different publications out there already. So these are just listing the advantages, and I'm sure the next speakers will illustrate those. Thank you for your attention.
Hello, everybody. My name is Anne Babayan, and I'm Cancer Research Marketing Manager at Illumina. And I would like to give you also some insights on the sequencing side of the things. So thank you again for joining, and welcome to another co-hosted webinar with NanoString and Illumina. Spatial analysis brings together the worlds of histology and sequencing, allowing us to study tumor biology at unprecedented depth and high resolution. This is done in situ on tissue sections, which allows us to interrogate a tumor complex biology in the context of its microenvironment. By pairing the resolution power of nanostrings genomics spatial technology with Illumina's leading next-generation sequencing readout, we can look at thousands of genes on the same tissue slide. These cutting-edge technologies are available now and are posed to revolutionize cancer research. High-throughput sequencers provide a large menu of applications critical for cancer research, from profiling of the tumor and its microenvironment using RNA sequencing at the bulk and single-cell level, as well as immune profiling using TCR sequencing and protein detection to analyze commonly utilized cell surface protein markers, and mutational profiles of the tumor using whole exome sequencing and whole genome sequencing. By partnering with NanoString, we are now adding another powerful tool to cancer research for cancer research to this growing list. And I think I'm experiencing some bad connection issues, so I'm going to switch off my camera just to save some bandwidth for to present my slides. So all our sequencing protocols, whether you are interrogating DNA, RNA, protein, or epigenetics, whether you decide to do any of those at bulk, single cell, and using spatial interrogation, basically consist of those four highly integrated steps. Sample preparation, as you can see on the screen, which is in the case of spatial analysis, consists of standard tissue sectioning protocols. The next is library preparation, where Illumina sequencing adapters are added, and subsequent sequencing and also analysis have been made simple and accessible as part of the workflow. Because of Illumina sequencers' data quality and throughput, Illumina sequencers provide the readout with which you can sequence up to 18,000 genes from a single tissue slide, interrogate the section at dozens of locations within the sample, and analyze dozens of samples in one sequencing run. Whether you decide to sequence on the highest throughput sequencer, the NovaSeq on the right, or on our newest mid-throughput sequencer, the NextSeq 2000, you can count to consistent data quality and the ability to analyze data across omics and resolution. What we offer is quite a bit of flexibility, and choose your sequencer depending on the amount of sequencing content you would like to interrogate. You can interrogate over 1,800 genes simultaneously with spatial resolution using NanoString's Cancer Transcriptome Atlas, or upscale your analysis with more than 18,000 protein coding genes with the whole Transcriptome Atlas from NanoString. And you can also add protein detection to look at known tumor cell markers. This is true multi-omics on a slide. In addition, to further streamline the workflow, in March we just announced public availability of NanoString genomics digital spatial profiling with Dragon on base-based sequencing hub. Read more about this workflow on our blog post. So today you will hear about NanoString and Illumina's combined spatial solution. Spatial biology is poised to revolutionize our ability to interrogate tumor biology, especially in a time when the understanding of the contribution of very complex tumor microenvironment is continuing to provide novel molecular targets for therapy, prognosis, predictive biomarkers, and diagnostic biomarkers as well. And spatial analysis and spatial multi-omics is already providing actionable insights, and some of which we will hear about today. And thank you for your attention.
Thank you very much, Rudy and Anna. Very clear and very, very concise, which is very good. So the next is going to be a short talk from Professor Noel De Miranda. He is associate professor at the Leiden University Medical Center. He's principal investigator of the Cancer Immunogenomics research group at the Department of Pathology of the Leiden University Medical Center. His group develops work on several aspects of cancer immunology and genomics, with a particular focus in colorectal and pancreatic cancer. The group's translational aims encompass identify cancer-specific antigens that can be targeted by immune therapy, elucidate potential of immune cell subsets other than T cells for immune therapy exploitation, and the possibility of understanding and mapping out the complex and multicellular nature of anti-cancer immune responses. Thank you very much, Noel, whenever you want.
Thank you, Ivan, for the kind introduction. I hope you can all hear me well. Good afternoon, everyone. I'm very excited to show you some preliminary work that we have been performing on gene expression spatial profiling to reconstruct colorectal cancer tumorigenesis. So I'm actually a PI of the immunogenomics lab, so our focus is slightly different from what I will show you today. Most of our work is based on the detection of antigens that are driving anti-tumor immune responses, but also the characterization of the complexity of tumor microenvironments. But throughout the years, we developed quite some expertise in spatial proteomics and genomics, and thereby we became very interested in making use of the genomics tool from NanoString. As you might be aware, and this is true for most cancer types, the survival of colorectal cancer patients is pretty much dependent at the stage of which a tumor is diagnosed, meaning that if a tumor is diagnosed at an early stage where there's minimally invasive disease and non-metastatic, the prognosis of a patient is much better than the one of a patient that is diagnosed with cancer that has spread to other organs, like in stage 4 disease. So national healthcare systems across the world have tried to address this by implementing early screening programs in the hope that one diagnoses cancer at earlier stages, and thereby the clinical management of those tumors is facilitated. So in the Netherlands, since 2014, everyone over 55 years old is invited to perform a fecal occult blood test. And as you can see on the graph on the right, we saw a huge increase in the incidence in the diagnosis of colon and rectal cancer after this test is implemented, which probably means an anticipation of diagnoses that were going to be done in later years. But what's very interesting, and you can see here in the second graph below, is that when diagnosis is done via population screening, the proportion of cancers that are diagnosed at advanced stages, at stage 4, it's much lower than in patients that do not undergo the route of population screening, showing that population screening really works in terms of anticipating diagnosis and facilitating treatment of colorectal cancers. However, this change in demographics of colorectal cancer accentuates other problems, which I would like to discuss with you on the next slide. So if you have a minimally invasive cancer, let's say a T1 colorectal cancer, you can be offered an endoscopic treatment rather than a full-blown surgery, which involves much lower risks and causes less morbidity. But if after clinical intervention unfavorable histologic features are found, or for instance, if you have positive resection margins, then the patient is recommended to undergo surgery. After surgery, however, it becomes apparent that metastases are in fact only found in 10% of patients that undergo surgery. So there is quite a lot of room to reduce the amount of surgery that is offered to these patients. On the other hand, there are also patients that are not recommended for surgical procedures and that are just treated by endoscopic procedures. And those do show tumor recurrences at a later stage, and we do not have good biomarkers really to identify the patients that are required to have surgery and the ones that are just really well off with an endoscopic procedure. So just to reiterate that, what we need really is good biomarkers that inform on the risk of metastatic disease. And we thought that the genomics approach, which allows us to assess gene expression across different regions of tissue, was really a great tool to address this question. And we thought that on top of having this translational question, this was also a great opportunity to redefine models of colorectal cancer onset and progression at the transcriptional level. So we applied the genomics cancer transcriptome atlas, which basically interrogates the expression of 1,800 genes related to tumor biology, tumor microenvironment, and the immune response on 8 different T1 tumors that were obtained by endoscopic submucosal dissection at the Department of Gastroenterology of the Leiden University Medical Center. And within each tumor, we selected 9 different regions that corresponded to regions of healthy mucosa, transition regions between healthy mucosa and dysplasia, low-grade dysplasia, high-grade dysplasia, and regions where there was very clear invasive cancer. And this to interrogate how this gene expression, whether we could detect specific gene expression alterations across these different histologies. And a very important detail, which is essential to refer, is that with the genomics platform, we were actually able to interrogate separately epithelial and stromal fractions. And we did this by employing antibodies marked with fluorescence that bound to pan-keratin and to vimentin. And keratin marks epithelial cells and vimentin marks stromal cells. And we were able to direct the acquisition of the specific acquisition of these regions so that we could interrogate gene expression separately in epithelial and stromal fractions. So in fact, for every region of interest, and I told you that we did 9 regions of interest for 8 different tumors, for every region of interest, we could distinguish gene expression profiles in the epithelial fraction and in the stromal fraction. And of course, in the epithelial fraction is where tumor cells are in the context of colorectal cancer. So we have employed the GeoMX suite, analysis suite for quality control, normalization, plotting of quality control and normalization parameters. But from then on, we exported the data to actually do our own analysis in R. And this project is being spearheaded by Jessica Ruland, a postdoc in the immunogenomics group. So in this slide, I just show you how these different samples separate or distribute themselves in the high dimensional space according to the overall profiles of gene expression. And what you can already appreciate is a near perfect separation of stromal and epithelial fractions, which is not really surprising, of course, because they comprise completely different types of cells. But it really gives you confidence that the method is working as you expect. But when looking deeply, and what's really interesting to see is that you also have a separation that largely agrees with the annotation that was made by histology. So normal and transition samples are close together in the middle of the TSNE plot, while there's also a separation according to the degrees of dysplasia or whether a region comes from an invasive area of carcinoma. And to really strengthen the argument that the biology makes sense, we overlay the enrichment scores for genes involved in Wnt signaling And indeed, we see a nice increase in the expression of Wnt signaling related genes as we move to the extremes of the TSNE plot, in line of what it would be expected regarding CRC biology and the involvement of Wnt signaling in cancer progression. So this is another way in which we have visualized the data, and this is the unsupervised clustering of the epithelial fraction alone based on the expression of all genes. And what you can see is that all normal samples clearly cluster away from the remaining samples, and that the majority of genes actually decrease as you observed malignant transformation. And something similar is also observed for the Vimentin positive fraction, so the stromal fraction. And what I find it really interesting to note is that at the moment, we have the feeling that as a whole, the gene expression patterns or differences observed in the stromal fraction appear to provide a better separation of the different histologies than the epithelial fraction. And this could be explained by the fact that there is large heterogeneity between cancer cells that are derived from different patients. But it begs the question on whether we can actually define biomarkers of tumor progression, not in the cancer cells themselves, but in the stromal cells that are associated with the cancer cells. So in these volcano plots, what you can see again is the differential gene expression stepwise as we progress from normal tissue to carcinoma, and as expected, there are small differences between normal mucosa and transition areas, and that is the graph on the left. But very interesting because in theory, these are the initiating events of tumorigenesis, of course. But once you really move into areas of dysplasia, the differences become huge between histologies. And this can be seen again in both epithelial and stromal fractions. So, as I alluded before, a lot of the biology makes sense, which makes us very confident on the methodological approach. So we see a gradual increase in the strength of Wnt signaling as we progress in tumorigenesis in both epithelial and stromal fractions, but also in other pathways like epithelial to mesenchymal transition, which is very closely associated to the activation of TGF-beta signaling, for instance. And this is really pronounced in the stromal fraction of the tumors, but also when comparing invasive cancer with other histologies. And this is, of course, very exciting because with the current approach, we know that we can identify biological mechanisms known to be involved in cancer progression in CRC, which really validates the approach. But what's most exciting is that we found several other pathways that behave in a similar manner, as I showed you for the Wnt signaling pathway and for the MT pathway, where you see an increase in activity of this pathway in both fractions, both epithelial and stromal fractions, but pathways that were not previously associated with colorectal cancer progression, and we're currently investigating and validating those, and I hope that we can share those with you soon. So far I showed you pathways that behave in a largely concordant manner between epithelial and stromal fractions of the same tissue. And this makes sense, right? Because a lot of these pathways, they're regulated by growth factors that have a paracrine function as well. So if a tumor produces a certain growth factor, it will not only impact tumor cells, but it will also impact stromal cells that are regulating, that are surrounding the cancer cells. But I want to zoom here on two examples of two pathways we found to be differentially regulated in the epithelial and the stromal fraction. So in the first pathway, which we call pathway M, what we see is a profound upregulation during tumor progression in the stromal fraction, but hardly any changes in the epithelial fraction, and if any, in the opposite direction. While for pathway D, we see an increase in the epithelial fraction during tumor progression, which is not mirrored in the stromal fraction. And these results really beg the question whether we would be able to pinpoint these processes as being associated to tumorigenesis if we had analyzed these samples by bulk RNA sequencing, for instance, where the contribution of the transcriptional profiles in both fractions would probably be averaged. I would just like to briefly mention one very interesting potential application of this undertaking, which is the easiest to translate, would be to identify individual biomarkers that can assist in the discrimination of healthy tissue from cancer, and which could, for instance, be used as therapeutic targets or biomarkers that assist in endoscopic or surgical procedures. And indeed, we identify in both epithelial and perhaps even more pronounced in the stromal fraction variations in gene expression that are strongly associated with malignant transformation and which we are currently validating. So the next steps for us will be we'll spend quite an amount of time at validating what we found at gene expression level to see if it translates at protein level. But also a very exciting component of this project is the intersection of this data with multiplex protein data that we have generated with imaging mass cytometry. And we are particularly interested in exploring the possibility to deconvolute the stromal expression signatures back to the cells that are composing that stromal component. So this was what I had to show you. Thank you very much for your attention. I'll just like to mention these people which are involved in the project, and of course, funding from the ERC and the LUMC, and I'll be happy to answer your questions later.
Thank you so much, Noel. Fantastic talk. Very well done. I'm going to ask the audience to make questions, and we will answer them together with the second speaker at the end of the second speaker presentation. But now we will run a poll. So you should select from your screen the appropriate answer, and we will share the results with you all shortly. So I will give some minutes for this. Okay, and now we can move to the second talk. Our presenter is Vasilis Stavrinides. He is a medical doctor who completed his junior surgical training in Oxford and London. He holds 2 postgraduate degrees in molecular medicine and mathematical statistics from the Imperial College and the University of London, and is currently completing a PhD on prostate cancer at UCL under Professor Mark Emberton, Professor Caroline Moore, and Dr. Hayley Whitaker. In 2018, he was awarded a clinical research training fellowship by the UK Medical Research Council for his research, which aims to explore the association between prostate tumor microenvironment, inflammation, and MRI phenotypes commonly seen in the prostate. Thank you so much, Vasilis. The floor is yours.
Thank you very much for the very kind introduction. Hello all. I'm going to hopefully share some exciting preliminary results from our work with nanostring in the next few slides. The title of the talk is The Transcriptional Evolution of Radiologically Progressing Prostate Cancer. You know already a little bit about my background because this was kindly provided. In terms of prostate cancer, we do know now that it's one of the most common cancers in men. Its incidence is increasing, and that's primarily due to the fact that awareness is increasing, and also that the introduction of prostate-specific antigen testing in the last 3 decades has basically led us to detect a lot more cases in recent years. Up until fairly recently, the mainstay of treatment for prostate cancer was surgery, or radical prostatectomy, that is, or some form of radiotherapy that was applied to the whole prostate. In later stages, the treatments consisted of chemotherapy and the administration of hormones. In recent years, however, these landscapes have slightly changed. There's now more modern treatments and some alternatives for very specific types of prostate cancers, which we'll see shortly. The important thing in diagnosis is obviously certification of risk at baseline, and that is traditionally done using a set of variables, the TNM stage, the PSA level, the cancer grade measured by the Gleason grade, which is gradually being supplanted by the grade group classification. And basically, with using these variables, we can divide roughly prostate cancers into those that have are relatively low risk and therefore pose a low threat to the patient in terms of the cancer-specific mortality, intermediate and high-risk cancers. This is an important exercise, clinical exercise, and particularly because there's preliminary evidence from previous decades that actually there are some prostate cancers that exist within the prostate that will never cause any problems during a man's lifetime. And there are others that actually are more likely to progress, evolve, and cause cancer-specific mortality, and those are the ones that need to be treated. And basically, up until recently, it wasn't that easy to differentiate the two. But clinical developments, diagnostic tests, and and clinical awareness has actually led us to a situation where we have to define those two different types, and also we have to be able to differentiate them when we are confronted with a particular patient. And this is truly important because, as you can see, the more aggressive and high risk the cancer is, the more it impacts mortality, and it impacts the recurrence of the cancer after it's been removed. On the left, you see a very famous paper by a figure from a paper by J. Epstein, European Urology, showing how the more high risk the cancer is, then the higher is the probability of biochemical recurrence after surgery. And on the right, there's some data from the very famous PROTECT trial showing that if you actually operate or treat with radiotherapy cancers that are not significant, your actual survival in terms of prostate cancer-specific survival doesn't substantially change. There is some improvement in the metastasis-free survival, but the actual overall cancer-specific survival, the prostate cancer-specific mortality does not change significantly. So as clinicians, diagnosing prostate cancer is not enough. We have to know which prostate cancers are significant and which are not. And that's something relatively specific to the oncology of the prostate. In other tumors, we need to know, we need to diagnose as early as possible, but simple diagnosis on its own in the prostate cancer context is not enough. We have to know whether the cancer is actually worth treating. And this is where active surveillance is a very exciting, established new way of actually managing those tumors that we diagnose, but actually are not, we're not very convinced that they do need treatment imminently. This essentially is a strategy where we monitor the patient over a period of time. We perform regular tests, prostate-specific antigen tests, sometimes biopsies, and we defer treatment until and unless there is evidence of disease progression. That essentially means that we can monitor patients over a long period of time, but we can only intervene when it is necessary, when we are actually confident that the cancer is actually more likely to lead to an adverse outcome. Surveillance cohorts are now maturing. There is some dropout from a lot of them, primarily due to disease progression, but there's also some non-adherence primarily due to the fact that a lot of standard active surveillance protocols now involve repeat biopsies, and obviously men avoid repeat biopsies as much as possible, so there is a little bit of dropout from that as well. The problem with standard sampling, prostate sampling, when we actually put a needle into the prostate to do a biopsy, is that, like I mentioned, the first problem is that the men don't like having too many of those. And the second one, up until fairly recently, was that the way that we did the biopsy in the prostate was fairly random. So we just deployed the needle in a few parts of the prostate without actually having any information beforehand as to how we should direct that needle in the prostate, and or whether there are specific areas we should be focusing on. And what that did is essentially introduce a little bit of a bias in our characterization of a given prostate. And in the surveillance context, as we saw, that can be detrimental, because what you don't want is to classify a high-risk tumor as low risk and put men on surveillance. This is where multiparametric MRI has changed management of prostate cancer very recently. By doing some imaging prior to biopsy, you can actually focus on suspicious areas in the prostate that are more likely to be significant cancer and sample those much more efficiently. At the same time, to kind of reduce the overdiagnosis of those insignificant foci of tumor that you do not really want to treat or are very unlikely to harm the patient in the long run. This is a slide, again, reiterating what I just said, that MRI has basically now revolutionized diagnosis of prostate cancer. It's advocated by the most associations in Europe and the United States. And at the same time, it appears that it can be a very useful tool for monitoring prostate tumors that are on surveillance, those significant tumors or intermediate risk tumors that we can actually monitor over time and defer treatment for. And what MRI does, it appears that it doesn't just improve certification at diagnosis that we just said, and therefore improves the selection of active surveillance candidates. But the other thing it does is actually can reduce the need for multiple biopsies during surveillance because we can use a non-invasive method to actually monitor tumors across time. Imaging-based cohorts are now they exist all over the world. One of them exists at UCL, recently described by one of our papers, as you can see here. And we have found that the more visible a prostate cancer is at baseline, the more likely it is for that patient to leave surveillance early and basically get treated earlier compared to other men who do not have, so let's say, visible disease at baseline. In those types of cohorts, we can actually monitor tumors and then only intervene and resample them when there is radiological progression. We do not yet know exactly what radiological progression is or what it's attributed to, and how we can properly define it. And the European School of Oncology precise criteria aim to do just that, to actually give some definition and structure when we are monitoring tumors over time with MRI to say, okay, which change is progression that we need to be careful about it, and which is probably just a normal variation over time. But we do know that MRI phenotypes are associated typically with microstructure of the tissue we're looking at. So therefore, could we have an idea of what's happening within an MRI lesion, a cancerous MRI lesion, as it progresses over time? Here, for example, on the left, you can see the image of an insignificant prostate cancer that is on surveillance. And on the right, you can see the same cancer after radiological progression. And you can see that the glands are more crowded, and tumor-infiltrating lymphocytes have actually entered the scene. And this is where perhaps the other thing you can see is actually this increase in cellularity post-radiological progression. And obviously, all these changes can be tracked back to some kind of molecular mechanism, and this is where the CTA from NanoString can be quite useful. In terms of the grant itself, the process was fairly easy. We had essentially tumors from men who whose cancer was sampled before and after radiological progression. These were FFPE samples. We sent slides to NanoString, used the workflow that was briefly mentioned, and this is the kind of data you would get back, typically thousands of genes, 1,800 genes from regions of interest. Very similar to the previous presentation, we can see that the first thing to note is that the assay could actually differentiate areas that were benign from areas that were cancerous. On the PCA plot here, as you can see, there is a very good differentiation and separation of the two. Even the areas where you see some overlap, actually a lot of them were high-grade dysplasia or areas that were not entirely normal. So overall, we could see that in the differentiation of benign versus tumor, the assay worked pretty well. The second conclusion is that the overall sort of gene signature in the benign tissue surrounding the cancer before and after radiological progression did not substantially change. Here you can see the top 50 differentially expressed genes, and at the top of the graph you can see blue for before progression and red for after progression sample of the same tumor. You can see that the transcriptomic landscape did not change significantly. However, when it came to cancer, the situation was quite different. You could now definitively say that there is a difference in the transcriptomic profile of a tumor before and after radiological progression. So I'm expecting to actually look at these results in a bit more depth and actually supplement it with a lot of other data. But in short, I wanted to give you a little bit of an idea what NanoString can achieve, the assay can achieve, and basically present to you the first preliminary evidence that actually, when you have the same prostate tumor sampled twice in time, that actually, whilst the benign compartment actually appears to retain its transcriptomic profile and characteristics, it is a tumor actually that undergoes the most profound changes in terms of its transcriptomic contexture. I would highly recommend basically using the NanoString assay. It was a very good experience. The team has been wonderful in assisting me through the whole experience. And like I said, I'm hoping to share further results with you in the future. I'd like to thank my funders, the ACR and NanoString, and obviously a whole bunch of people, but there's just too many to thank, so I'll just pass the floor to the next person.
So thank you so much, Vasilis. Fantastic talk again. So let's proceed now with the questions. There are several of them. I will ask the speakers. I will also ask them to be short because there are many questions, so we can go through them quickly. So let's go for the first one. This is a question for Noel and Vasilis. What was the major advantage for you to be able to select the regions of your interest to dive into, and also to look at different tissue compartments with those regions?
I can go first. So for us, the major advantage is that we're very much interested in looking at the cancer cell compartment and the stromal compartment separately, which also stems from our interest in tumor immunology. And of course, the ability to look at different tissue compartments allows us to correlate gene expression changes with specific morphological alterations that we can see in the tissue, or that the pathologist can see in a tissue to see whether they stand, or if we get some more information from gene expression.
Vasilis, do you want to complement?
Yeah, I think definitely pathology. I agree with Noel. It's basically pathology is essential when you do these kind of projects. You want to identify areas of interest and then stratify them according to the severity or the grade of a particular tumor. Identifying dysplasia, for example, can be notoriously hard for the non-expert. So I would definitely recommend having pathologists on board the journey. Beyond that, for us, as you saw, the main thing was to actually the main purpose of the project was to actually try to decipher how the same prostate lesion evolved over time transectomically. And obviously the needle is never perfect. It goes a little bit to the left, a little bit to the right. So being able to capture all the heterogeneity from a single sample was essential to actually start drawing some comparisons, because otherwise if you did bulk work, you wouldn't be able to tell what's the main driver behind that transectomic change you see on the data.
All right, thank you. So this is a question to Noel. Is it possible that population screening detects tumors that would never become clinically manifest? If yes, do you have any idea based on your research which one of these could be? Probably the same discussion applies for prostate cancer.
Yeah, I mean, that's a very good question, but it would be very difficult to predict which tumors we are analyzing, or which ones would be the ones that would not develop into full-blown cancer. Of note, we did only include tumors that clearly show an invasive portion, so clearly classified as T1. But I'm sure there would be a number of these tumors that would go asymptomatic and not be diagnosed otherwise. I'm not really sure how to test that from a methodological point of view, though, because once we remove it from the patient, we don't know anymore whether that tumor would develop or not to full-blown cancer.
Yeah. So, Vasilis, do you want to say something about this?
Yeah, I think, like you very correctly said, it's. I think with prostate cancer, where it's a bit of a different landscape in the sense that you have a blood test that has to be investigated. So as soon as you sample the tumor, then you have to make a clinical decision. It's a little bit easier compared to other malignancies in that you know, for example, once you see a low-risk tumor, that you do not necessarily have to treat it for using outcome data. But I agree that the main thing is that we have a blood test, and the majority of them are asymptomatic, whereas in colorectal cancer, unfortunately, you have the disadvantage that a lot of the cancers are picked up at a symptomatic stage, so there is no option for surveying those, and monitoring those cancers is not an easy endeavor. So, but in the prostate cancer, we definitely have a problem over diagnosis. We're now picking up way more insignificant disease that we would like, and that's where MRI basically is a little bit helpful in reducing that burden to the patient and the healthcare system.
Perfect. Let me chip in, and I have also a couple of questions, one for each of you. First for you, Noel. Have you seen differences between different subtypes of colon cancer, such as CMS 1, 2, 3, 4? Did you found some differences, especially in the stroma for CMS 4?
Yeah, so that's actually something we would be very much interested in trying it out to see if we can translate the algorithms that classify the CMS subtypes across the spatial contexture. That's definitely something on the list. It would also be nice, perhaps, to see whether there's heterogeneity within the same tissue regarding the molecular classification based on RNA expression. Yeah, that's a very good question and suggestion.
All right, and for you, Vasilis, I noticed that in your example there is an increase in T-cell infiltration. I mean, is this one of the differences you are finding in your cluster that is increasing cancer in cancer development? Do you see more immune infiltration?
Well, that's actually something we're looking to do next, because you would need ideally to quantify that somehow, and that objectively can be done with things like multiplex immunofluorescence, or actually look at the H&E images. So we are looking into doing that, but it's definitely interesting to see not just the quantity, but also the qualitative aspects of what kind of inflammation do you get, because you might get inflammation in the early stages, which might be associated with the benign or premillennial process, but that doesn't necessarily mean that because you say an infiltrate later stage, it's the same infiltrate. It might be different in other ways. And I think that's where, for example, nanostring could be useful in deciphering and decomposing basically exactly what's going on in a specific area. In terms of the benign signal, it appeared, and we have a very small sample, it appeared to be the same before and after progression, even after the cancer grade increases. But I wouldn't necessarily, hold on to that result just yet, primarily because the sample is small. But even if it were to be true, it's very useful because it points us to the direction of where we should be looking. So maybe expanding the transcriptomic signature, looking at other types of signatures is also very clinically useful, which we wouldn't have done without special resolving these men. So yes, there are plenty of other applications we can look into.
Perfect. Let's go on with the questions. The other question is for Noel. How long did it take you to generate these great results from cutting the section and to getting and analyzing the final results? I guess it's a very technical question. I don't know if you want to answer that.
Yeah, I mean, we still didn't get to the final results, but maybe the question is focused a bit more on the wet lab part and the selection and data acquisition. That was actually quite fast. We planned a session with the nanostring people, and that went very smoothly. It actually took us longer to organize which samples we were going to process and get them cut than acquiring the data. But then, of course, you get a wealth of data, and you run the risk of being mesmerized with it, exploring all different things that might be in the data. So at some point, we will have to say stop and just release it for everyone to have a look at it.
Perfect. So a question for Vasilis. Do patients prefer MRI-based surveillance over standard non-imaging based protocols?
It appears that roughly speaking, it appears that they might do. I think as I briefly mentioned in the lecture, one of the primary disadvantages of standard protocols that are not imaging based is that you have to do biopsies every now and then, and a lot of protocols are now quite prescriptive, and obviously men do not necessarily like that. So you do see a substantial drop after 3 and 5 years in terms of their compliance and their anxiety. But if you use imaging, even if you spare them from a few extra biopsies, actually that seems to improve their compliance. We had very little people dropping out at UCLH, although it is a quite specialist environment, but still we had very few dropouts. And it appears that from my clinical experiences, basically that people are really involved. They like looking at an image, they like looking at their tumor on imaging and seeing how it is. They just seem more involved with it rather than just presenting them with a pathology report. So that helps. But obviously, it would be good to study what's a long-term effect of introducing imaging in the surveillance pathway and see exactly what's going on. But the evidence is definitely for it rather than against it at the moment.
Perfect. And now for you, Noel, would you consider subdividing the data acquisition process for additional cellular fractions in addition to keratin and vimentin?
Yeah, so I think there are a lot of possibilities depending on what you're interested in. For instance, we could have thrown into the mix also an antibiotic against CD45 and further subdivide our fractions into epithelial, stromal, non-immune, and immune. I think it's just a matter of having a good antibody and an antigen that survives the procedure.
All right, just 2 last questions. For you, Vasilis, how often do prostate cancer on active surveillance progress radiologically and pathologically?
Radiological progression is not a common event. It does happen ultimately to many men, but it's not a common event. When it does happen, the real question that we're confronted with is basically whether we should sample that lesion again, whether it reflects true pathological and disease progression, or whether it's just normal variation of an imaging phenotype over time. And it appears that from a very preliminary analysis we've done is that roughly 2 out of 3 times, so about 60-something% of the time, if you have radiological progression on imaging, you will get pathological progression on biopsy. So MRI seems to be pretty good at picking up those changes, those microstructural changes that are accompanied with increase in grade, cancer grade, or the growth of a particular cancer. Like I demonstrated from the example that I showed you, is that you do get a change in the transcriptomic sort of contexture of the tumor, of the same tumor at 2 points in time. It changes in terms of what genes it expresses, whereas the surrounding environment seems to be relatively stable, although very preliminary data need to be supplemented with lots more, but that's what I showed for now.
Perfect. And the last question for you, Noel. Can you say something about the potential clinical application of a marker that you determine to be associated with cancer progression?
Yeah, so I hinted towards that at the end of the presentation. If we can use biomarkers to help improve staging or predict the occurrence of metastasis, for instance, that would be great. But we also have, for instance, in the institute, image-guided surgery departments. If you actually find a marker that can be translatable to a probe to be used in image-guided surgery to actually have a surgery where you're able to remove the whole tumor, that would be a potential application, another potential application for a biomarker.
Fantastic. Thank you so much, both of you. It is a pleasure for me to hear you, and I think it's been a fantastic session. On behalf of EACR, thank you for being here and presenting your data. And now, just to finish, I want to go on and as pre-announced, inform you about the great opportunity to apply to the EACR NanoString and Illumina Oncology Grant. We will award 2 winners who will receive a free of charge whole transcriptome project for the human or mouse tissue, FFP, or fresh frozen tissue. We ran a similar grant last year, and it was very, very successful. And so we wanted to repeat, open up up to abstract submission from today till the 24th of May at midnight. The winners will be able to send 4 slides of tissue to Seattle, where the R&D team will run them on the GeoMX and NextSec 2000. The winners will receive back the raw data as well as the results after data analysis, and of course, free of charge. And so you can learn more getting into the websites where you will see all the information about that, and I think it's going to be shown in the presentation right now. So again, thank you very much for this time, for this fantastic session. And there is also a last poll that I think you may want to run and fill it. And again, thank you all, and see you next time. Thank you.
After completing slide preparation, which is similar to IHC or ISH, we will put our slides into the tray, and then we'll put the tray into the instrument and load buffer. Next, we will be ready to enter our slide records and scan parameters. A fluorescent image with up to 4 colors is generated to enable selection of region of interest or ROI on this tissue. You as the user can choose these ROIs from anywhere on your laptop. Geometric profiling allows us to choose circles, squares, or even polygons. This would be a great tool when you have pretty homogeneous regions like immune cells and the tumor segregated within one tissue or across tissues in your cohort. Geometric profiling was utilized in David Rimm's Nature Communications article in 2020. Many users can also create rules for segmenting the ROIs. This would let morphology of the tissue determine the boundaries of the area of illuminations, or AOIs, to enable 100% cellularity. For example, users can create tumor and TME segments across a tissue sample as shown on this example. Selected ROIs are illuminated with UV light to release barcodes from DSP probes. And then these DSP probes are deposited into a 96-well plate. The released DSP barcodes are prepared via a standard library preparation, so they can be read out on Illumina sequencers. The data can be mapped spatially back to the originating region of interest. Samples can now be loaded onto your Illumina sequencer. Depending on your experimental design and the instrument capacity, you can read up to 760 ROIs from a single genomics experiment in a single Illumina run. Here we have an XE 2000, which will allow us to easily read hundreds of protein or RNA analytes. Once the FASTQ files are generated, they'll be converted to digital code counts and then uploaded back to Genomics DSP instrument. The data analysis screen is divided into 3 panes and a taskbar. Here we describe the functions of each pane and the interactions between all 3. The scan pane is shown on the left side and contains individual viewers of the scans. In the middle pane, we have our data sets and probe lists. On the right, we have our visualization pane, which shows the various graphs. In this case, this is our QC graph. The middle datasets pane lists all datasets and probe groups associated with the current study. The visualizations pane is on the right. Visualizations are visual representations of the dataset selected and probes selected, and the adjustments applied to the dataset from those probes. Now I'm going to open another study. Here we can see the data has been QC'd and normalized. Once you normalize the data, you can see in your visualizations pane different graphs, including pathway maps, pathway tables, bar graphs, correlation plots, box plots, PCA plots, trend lines, and we can view heat maps. I'm going to end with clicking on a data point on the data visualizations pane, and you'll notice that ROI associated with it will be zoomed in on the left pane of the screen, presenting seamless integration of the images and the digital counts. Thanks for joining our Customer Experience Lab sessions.